Hello, in this video we will discuss about the neuromuscular junction is the also discuss about the sarcomere contraction as a sliding filament model it is also known as. So let's we will discuss and recap the spinal cord to understand the neural pathway, a nervous system pathway which that is also understand for the neuromuscular junction. So here is the spinal cord, which that is contain a upper nuclei and basically is the anterospinothalamic tract is the term. So here is the higher somatosensory cortex, which that is detect the some pain, pressure or touch via a afferent nerves. So here is the dorsal ganglion. This dorsal ganglion is the sensory nerve fiber for the detection of pain, touch, and pressure, nociceptor or proprioceptor. So in this way, here is the ventral ganglion. It is contain a motor neuron. So here is a anterospinothalamic tract, which that is basically we will discuss also for the motor neuron. So the sensory neuron will sense the stimulus, while the motor neuron will give response in the form of afferent nerve. So here is the ventral horn of gray matter and dorsal horn of gray matter is the butterfly-like structure, while the outer is the white matter. In the case of brain is the opposite this. Basically the gray matter is opposite. So lateral path, lateral side. So let's next here we will discuss about this is the hand, and after the needle will give the response of the pain. So in this way the dorsal ganglion will contain a afferent nerve, sensory nerve, which that is transmitted through a anterospinothalamic tract, and in this way the motor neuron will give response after the integration, and in this way the ventral ganglion of motor neuron which that it gives the response by the neuromuscular junction is here. So in this way the thalamic nuclei which that is interconnect and central relay. So in this way here is we will just understand the neuromuscular junction. And what is that? Neuromuscular junction is the junction between the muscle fiber with the nerve. So this is the nerve ending, which that is a myelinated. It means it is the motor neuron. And myelinated means it the nerve impulse will be quick as compared to the unmyelinated cell of the nerve. So here is the synaptic cleft and it is the nerve ending. It is the knob-like structure, and it is here is the potassium sodium exchange to create the action potential to create nerve impulse. So this nerve impulse is the saltatory conduction. Why? Because the Schwann cell are present. It is the myelinated. So the myelination is occur, and node of Ranvier will transmit the again further transmit inside. So you can see the nerve impulse will move from inside. So it is we can say the jumping mechanism of the nerve impulse, which that is depolarization further and further to quick response. So the calcium move inside by the depolarization. It means the calcium, extracellular calcium will move inside. And when extracellular calcium will move inside to stimulate several things. Here we will just understand the a little bit amount of choline will move inside through a high affinity choline transferase channel. So channel is enzyme-like, and choline will move inside on the base of here is a lot of mitochondria near the knob of the nerve ending. So here is acetyl CoA will be coenzyme will be released from the mitochondria during the aerobic respiration. So the choline esterase enzyme used to acetylcholine formation by the binding of the choline with acetyl CoA, and in this way the calcium are present already, which that is create the exocytosis of this vesicle containing acetylcholine containing vesicle. So this is the exocytosis. It means the releasing of the acetylcholine. And after the releasing of the acetylcholine will bind with the acetylcholine receptor. And near the receptor is contained a channel, sodium voltage gated channel. The sodium voltage gated channel will depolarize after the move influx of the sodium ion. So here is the positive charge due to the sodium cation extracellularly, and here is the intracellularly potassium cation which that create the negative charge. And there are many type of protein which contain a negative charge, so that is why it is negative charge. So this negative charge will be converted into the outside is the negative and inside is the positive, is known as the depolarization through a sodium influx. So influx of the sodium will depolarize, and this nerve impulse will move toward the T tubule. T tubule is the basically the muscle fiber contain a T tubule. So the muscle cell basically contain a T tubule, and it is the sarcoplasma. The sarcoplasma is the plasma membrane like structure. We can say that sarcoplasma because it is the muscle cell. So in this way the sodium will stimulate the DHHPR channel to calcium influx, and the RYR1 which that is allowed to move in flux of the calcium. So in this way the calcium will stored into the sarcoplasmic reticulum. Sarco means the contraction, so the reticulum is the like a endoplasmic reticulum. So contain channel, calcium voltage gated channel. It means the increase of the calcium will stimulate the stored calcium outflux into the sarcoplasm. So when you move into the sarcoplasm, efflux of the calcium into the sarcoplasm will bind with the sarcomere where the actin and troponin is present. So the tropomyosin also we will discuss also. So the calcium when bind with the troponin to begin the myosin head will contract. So this is the sarcomere, is the unit of muscle contraction and made up of actin and myosin. And here is the Z to Z line is known as the one sarcomere, and anisotropic and isotropic band. Isotropic means the light can pass from it, while the anisotropic light will not pass. So the H zone is between the gap where the muscle contract. So that is the central axis. So let's begin to understand the relaxed state, cross breach, and relaxed state. Again, that is the antagonistic work. We will also understand what is that. And here is the relaxed state. First of all, we will understand. So during the relaxation, the actin and myosin will relax and not attach to each other. But when contract, that means the before the contraction will bind with the actin with myosin. So first of all here we will discuss about this is the myosin filament. For myosin filament is also known as the thick filament. It is made up of tube and myosin head. Pair of head are present which that is the contained with a tube or some you can say that is protein containing myosin. So this is the myosin, it is the protein. So here is the actin. Actin contain basically troponin as well as tropomyosin thread like structure, as well as the tropomyosin and actin. Actin is a bead like structure, and troponin is also bead like structure, is the trimeric part of the protein, with the each groove contain a troponin. So in this way you can see that is the calcium when bind with the actin. So when calcium will bind with the actin, so that basically is that myosin head is the basically dimeric protein. Here is that the actin is the thick filament and myosin is the thin filament. Myosin is the thick filament and actin is the thin filament. So it is the sarcoplasm. The myosin and actin are present in the sarcoplasm of the muscle cell. So the muscle cell is a multinucleated, not anucleated like a smooth muscle, but the skeletal muscle. Here we will discuss about the skeletal muscle. So the skeletal muscle is basically is striated and with a multiple nuclei like a cardiac muscle cell. So I hope you make sense these things. So Here is the relax when it means the relax means the contracted tricep, but the relax biceps. So the tricep will be contract, the bicep will be relaxed. In this way, it is known as the antagonistic work of the muscle contraction. So one muscle will be contract and the one will be relaxed in this manner. So the cross bridge will be occur. Cross bridge means the actin and myosin head will be bind together is known as the bridge formation. And this bridge formation is basically the myosin bind due to the calcium will attach with the troponin will stimulate the binding of the myosin. After the myosin will be bind with the actin filament actin filament. So the troponin, where is the troponin are present, so the myosin will be bind. After the binding, the muscle contraction. So in this way, the muscle contraction will be occur. It means the move in the center of the H zone. H zone is the sarcomere which contain. We will discuss also to understand. Just you should think about that. It will move toward the center. So in this way, the shorten of the muscle. So here is the, for example, relaxed state, which that's contain actin and myosin filament. Black color shows that the actin filament, while the red color is showing that the myosin filament. And the muscle cell also contain a myoglobin. This myoglobin is contain high affinity of oxygen absorption from the blood circulatory system. So the detachment of the oxygen from the hemoglobin will be less affinity as compared to the myoglobin. So in this way, here is the sarcomere Z to Z line and the isotropic band and anisotropic band and H2 zone are present. So the more anisotropic gap due to relaxation of sarcomere. So the Z to Z line and isotropic band is here. So you can see during the contraction, here is the contraction time. So when contraction, the H zone will be decreased. So the H zone will be decreased and isotropic band will be more prominent, and anisotropic band shows more during contraction of muscle. So the anisotropic band will be prominent. In this way, the microscopic study will show this all process. So just remember about that. On the other hand, the relaxed state we will discuss if. So the basically when relaxation require ATP. So in this way, we want to say that is the ATP convert into the ADP by the hydrolyzation of the myosin. So the myosin will bind with the phosphate group to hydrolyze and the calcium will be detached from the troponin. So the creatine is also important, which that is extra store of the phosphate group. This is look like the ATP, which that provide the energy for the detachment. So if the excess of ATP are present in the muscle, will store in the form of creatine phosphate. So here is the relaxed state. In this way, the relaxed state we will discuss the ATP is provided from the mitochondria present in the sarcoplasm, and this ATP will be bound with the myosin head to hydrolyze this bridge, and it is the cross bridge will be hydrolyzed and the calcium will be released and troponin will be free, and the actin myosin tropomyosin and actin filament will be detached and relax again, and again tricep contracted and bicep will be relaxed. So in this way, the antagonistic work will be occur. So the rigor mortis is the after death ATP deficiency lead to relaxation of muscle and drastically decrease and not relax to relax muscle is not able to relax muscle is known as rigor mortis after death. So important thing is that the early death phase will able to contract or relax with the help of other person, but the time wise will be rigor mortis will be occur. So the muscle fatigue is that during high intensity exercise create a less amount of oxygen to myoglobin and glucose breakdown not complete and lactic acid produces and calcium level also interfere in this way. The decrease of the calcium level will cause the muscle fatigue. So in this way the muscle